Hey, what's up everybody, and welcome back to the Sanctuary. I'm your host, Professor C, and we're going to do some more A&P today. Specifically, we're going to check out a little more neurology. We're going to talk about the synapse and the process of neurotransmission. So, let's check it out. So we have to remember a little bit about our previous talks, and I'll kind of sketch out a neuron really quick, kind of down and dirty style. We've got a cell body here and an axon leading away to some sort of terminal button, and we'll just say there's only one axon terminal on this particular neuron. Now, when we talk about neurotransmissions, neurotransmitters have to come from somewhere. They are going to be synthesized or created here in the cell body, and they're going to be shipped down to the terminal knob where they will be stored until they are released, is what it says in this first statement. Now, there is a space here where it touches, and we use that word very loosely where we say touches. There's an actual space here in between, and sometimes you hear that called a synapse, the actual connection there that's not a connection. Sometimes you hear it called a synaptic cleft. So there is going to be a gap in between here, and if you studied my lectures during the neuromuscular junction activity, you saw the same thing, separation by a little bit of a space. So it's not just an empty space filled with air, it's filled with fluid. And one might ask, if you're going to do neurotransmission, why not just plug in one neuron to the other the same way that you might plug in, say, a cord into a socket into the wall and get some electricity to flow? Well, the main reason that there is a space is to prevent directly passing a signal to the next neuron. And you may again say, why not just plug it in? Well, this is to ensure that we only get one directional transmission, right? One way only. We can't go backwards in the transmission because of this system. So whenever we have a neuron, what we say firing or shooting off its neurotransmitters, and in this case the image is shooting them that way, we would call this a presynaptic neuron. So I'm just going to write the word pre here on top of that neuron. And it is shooting its neurotransmitters, the small balls of material that are moving across that gap, is going to be called the postsynaptic neuron. So we have a pre and a post. And very cleverly, in some textbooks, you'll see this referred to as neuron number 1, and postsynaptic neuron will be called neuron number 2. Now, these are chemical synapses. There will be some other tricks we'll learn a little bit later. But because these are specialized to create and store and release and receive neurotransmitters, these are chemical types of synapses. So we're going to, again, create, move the neurotransmitter down to the terminal end of that neuron, the presynaptic neuron. We're going to put that material into vesicles where they will merge with the membrane and diffuse across to the postsynaptic neuron. Sometimes that postsynaptic neuron can be, say, a muscle fiber. If you say that looks like a neuromuscular junction, it could be in this instance if the neurotransmitter were acetylcholine. So another kind of difference here in the nervous system, whenever we saw a neuromuscular junction, if we did ship neurotransmitter acetylcholine over to the muscle, it always excited that tissue. We may now see inhibition of the second neuron. The postsynaptic neuron may be inhibited by the release of the neurotransmitter. Now, to help this process, we have some docking proteins. And what we have to do first of all is realize that these action potentials that we created, maybe up in the cell body or even beyond, hit the knob at some point. And this is what it says first. When nerve impulses or action potentials hit the terminal knob, they will cause an influx in calcium. Voltage-gated channels will open and calcium will rush into the cell. And this will turn on some special proteins called docking proteins like synaptotagmin and some of its cousins. And they will help package the neurotransmitter and then push it out of the cell through exocytosis. Now, there are a lot of neurotransmitters that we have discovered. Some of them arguably are called hormones in other chapters, and at some point I think we're going to have a neuroendocrine system just merged into one and talk about neurohormones. But as it stands now, it depends on what chapter you're in, whether oxytocin is called a hormone or it's called a neurotransmitter. So most neurons make at least two, sometimes more neurotransmitters, and they are released depending on what type of stimulation frequency they are receiving. Over 50 have been classified and categorized into different groups. And we're going to take a quick look through some of the major groups. So 7 classes, these aren't the 7 classes of neurotransmitters. You can group these in many different ways. I'm just going to pick 7 classes that you often hear about, which are acetylcholine, glutamate, Gaba, also known as gamma-aminobutyric acid, dopamine, adrenaline, also known as epinephrine, it's the same thing, serotonin, and oxytocin. So let's briefly check out each one of these. So acetylcholine is something we've seen before. If you've watched my talks, we talked about it a lot when we talked about muscle activity, because this is the neurotransmitter that's released at neuromuscular junctions. We'll find out a little bit later. It is released at some ANS neurons here. I'll circle that, autonomic system, right? And we haven't talked about it at all yet, except to introduce it. But we'll revisit this acetylcholine idea when we get to the autonomic nervous system. So it's synthesized from an enzyme called CHAT, choline acetyltransferase, and it basically glues together acetyl-CoA and choline. Later on, when we need this effects to be terminated, we will use another hormone called ACHE, acetylcholinesterase, which breaks it back apart into an acetate and a choline. Glutamate is very common in the diet, and it is by far the most abundant excitatory neurotransmitter in the brain. That is, it has excitatory action and it likes to turn on other neurons. It plays a huge role in learning and memory. It functions as a neurotransmitter in almost every type of animal except very, very few, including very simple animals like the comb jelly you see next door use glutamate as a neurotransmitter. It's also been linked to problems like amyotrophic lateral sclerosis, phenylketonuria, Alzheimer's disease, and some other problems. GABA, simply, or you could say it complexively, say gamma-aminobutyric acid, is another very popular neurotransmitter, and it's pretty much the polar opposite of glutamate. This is a chiefly inhibitory neurotransmitter in vertebrates. And just as a side note here, insulin-producing cells that are called beta cells in the pancreas, they inhibit alpha cells which produce glucagon, which is the antagonist to insulin, and thereby they stimulate their own growth. So you will hear about GABA not only in the nervous system itself, but also inside of the pancreas. Dopamine, everybody's heard of this one before when you talk about getting that dopamine rush of pleasure. And it is true what people say, but it plays a lot of roles in the nervous system. So in the brain, dopamine plays very important roles in what we call executive functioning, like broad motor control, motivation, arousal, learning, reinforcement, and of course pleasure and reward, as everyone has heard about. But it also does some lower level functioning as regulating appetite, lactation, sexual gratification, and in some instances causing nausea. Adrenaline is one everyone's familiar with. It is also known as epinephrine, and there are the cousins noradrenaline, also called norepinephrine, but they all do the same thing no matter what their name is. They play this role in this fight or flight. Sometimes I describe it as fight, flight, or fright response because it could be something you're not actually going to be hurt by, but it scares you anyway. So this does a lot of things, and just imagine getting ready to either be in a fight or run away. You're going to have more blood flow to your muscles, your heart's going to start beating faster, you're going to increase your breathing rate, your pupils will dilate so you can get a lot of light to see where to run, your blood sugar will increase so you can get a lot of really raw power very quickly. Serotonin, another one that has to do with broad regulation of behavior and mood. So this is a neurotransmitter, a lot of complex functions, but learning, reward, memory, and cognition, these are things associated with serotonin, as well as some other things like vomiting and vasoconstriction, that is squeezing of the blood vessels. Interestingly enough, 90% of serotonin is not produced in the brain, it's produced in the GI tract where it regulates intestinal movements. One of those little side notes that pops up on standardized exams sometimes and gets people, but it is true. Oxytocin, as you can tell by the picture, has something to do with bonding in some way. And sometimes this is called a hormone. When we get to the endocrine chapter, we will talk about oxytocin, or OT, as being a hormone. But in this chapter, it's referred to as a neurotransmitter. So it does play a role in behaviors that are very complex, like social bonding and pair bonding, and then getting into reproductive habits and actually childbirth and post-childbirth bonding periods. Synaptic delay is a term that's pretty self-explanatory. In order to have this happen, neurotransmission that is, you have to release a neurotransmitter. It has to diffuse across the gap. It has to bind to receptors on the other side, and this takes a little bit of time. The key word is a little bit of time. Average synaptic delay, depending on where you are, is anywhere from 0.3 to 5 milliseconds. And of course, at some point, you want the transmission to be terminated. You want the end of it to happen. You don't want to have continuous transmission. You want it to stop. So within a few milliseconds, of course, neurotransmitter effect is terminated by a variety of mechanisms, such as enzymes like we talked about with acetylcholine esterase breaking down acetylcholine. So we can have just natural degradation by enzyme. We could have a thing called reuptake happen. Either astrocytes can do this, or sometimes the actual terminal of the axon itself can reuptake any extra neurotransmitter that may be in the gap. And then, of course, because there is a gap, there is a synaptic cleft in between the two neurons, sometimes this neurotransmitter can simply fall out of the cleft and diffuse away. That's why you release a lot of it when you do, so we ensure that we get the signal across the gap. All right, hope you enjoyed that little talk on synapses and neurotransmission. Thanks for watching it to the end if you did. Check out some more videos in my series if you want to learn more about A and P. See you for the next one. Bye-bye.